Good day everyone, welcome to the course on biological data analysis and visualization with R. In the last few classes we have looked into chip-sec data analysis, we have looked into the workflow, we have started the hands-on where we have started with BAM files where we had the actual treatment data, right, the H3K specific methylation data as well as the input data. So, we have followed the steps, right, where we have first converted the BAM files to bed files and then we have done specific analysis on that data. We have chosen only chromosome 3R because of our gene of interest. We have then loaded our annotation files using ensemble biomart, right. Biomart we have accessed the ensemble data and specific version of the genome and finally we did the binning and that is where we ended the last class. Okay. So, in the last class we have used the binning function, right, where we actually get the count of reads per bean of the genome, right. So, we divide the genome into different beans of specific size and then we count how many reads fall into those beans. Okay. So, what we are going to do is we will take that forward and we will start with something called peak calling and then we will look into the visualization. Okay. So, we will see how these beans actually look like, we will visualize them. We did some very basic visualization in the last class where we plotted the read distribution around our gene of interest, but we will see much more sophisticated visualization techniques in this class, where we will see, for example, the location of the genes at where these are located, their orientations, etc., and then the counts or the density of the reads, etc. And then we also can overlay the peaks once we have done the peak calling part on those visualizations. Okay. So, again just to remind you, this is where we are, right. This is the flow chart. We have started with BAM file, we have worked on bed, right, we have converted that to bed and then we have done this binning already in the last class. We have used the package chip-sec and then what is remaining is the peak calling part and the visualization part. So, for visualization we are going to use these packages RTrack layer and GVs. So, we have already loaded these GVs and RTrack layer packages and this is what we are going to do in this class. So, for visualization, as I said, we have this genome annotation. So, first thing we will do is generation of data tracks, okay, with the data that we have and then we will plot these data tracks with counts and peaks. Okay. And the genome annotation that we have already loaded using biomart, we have that information. We have the feature map. Again, if you go back to the last class, we have loaded this feature map. For the data tracks and the plotting, we will use these packages RTrack layer and GVs. Okay. So, with that overview, let us now go into the R console and we will do the hands-on part, right. So, and then I will first remind you this binning part that we have done already and we have seen some of these visualizations and we have exported this data in the bedgraph format. Okay. So, again we can load this bedgraph data and we can visualize if required. Anyway, so we have this data. So, for visualization, so we will first start with this visualization of this read data or the count data. Okay. And for that we need this biomart, right, where we have this biomart database for Drosophila gene annotation, right. So, we have this gene information there. So, the first command is we need to define a genome axis track. Okay. So, this is again something that we are going to use for our visualization. What we can do, we can test these different visualization. For example, you can start with this function plot tracks. Okay. And what we are saying is with this genome location, right, genome axis, also use this metry location, right. This location we have already defined and this want to plot this region. Okay. Transcript annotation should be ensemble transcript ID, window is auto and then these are some specific parameters for controlling the size of the title and the font size, etc. Okay. So, let us start with this plot tracks function and see what we get. Okay. Right. So, this metal 3 location, this is our gene of interest. Okay. And this actually has stored the information. Okay. So, what you see here, right, we have this information now plotted, this genome axis track, this is chromosome 3R, this is the location. I hope you can see this. Of course, the font size is not too big. This is the location in the chromosome 3R, right, 24 MB. This is the location, and this is where our genes are present, right. Many genes are present. So, our gene of interest, let us look at the ID, right. This is FBGN 0039139. Okay. So, this is the gene. So, our gene of interest is here, this is the one that we have. Okay. All right. So, this is the gene that we have, it is present right here. Okay. We can of course increase the interval, or we can also decrease the interval, so that we just focus on our gene. Okay. So, for now let us keep this interval, right, genomic region that we want to look at. What we are going to do now is first define these tracks, data tracks. Okay. So, this data track, the information will come from these beans files, right, so where we have done the binning, and this is the first command. We are defining the control track, right. So, we want to see, right, how this read count is changing, all right, between this control versus the actual experiment, right, where we have these histone modifications, where we have done the ChIP-seq. Okay. So, the control track first we need to define here. Okay. We will use this binning information after we have done the binning, right. The genome version would be DM6, the histogram color would be gray, right, field histogram black, name should be control, color axis should be black, and then there are some parameters. Y limit is 0 to 2000. So, this is something that is important. We need to define the Y limit, same for all the tracks that we plot. Okay. So, this is something I have plotted, and I know that 2000 will give us a nice range for visualization of these results, but this is something that you can play around and change. Okay. So, let us first define this control track. Okay. And then similarly, we will define control track, the tracks for the ChIP-seq experiments, the replicate 1 HTK36Me3 replicate 1, right. This is the replicate 1 track, and then we have the replicate 2 track. Okay. Okay. So, similar way we have defined these tracks, right. We have used this beans information that we have generated in the last class. Okay. So, once we have this, right, we can now plot these tracks, and we can look at the read distribution or the coverage across the genome, and the command is plot tracks. Okay. So, what we will want to plot is these 3 tracks, right, the control track, the replicate 1 track, and the replicate 2 track. Okay. We plot these 3 tracks, okay, along with the location of this gene and the genome axis track GAT. Okay. So, GAT and METL3 localization, right, this will give us this plot again, right, but we want this along with the binning information, right, what is the count data for each of these samples in this specific region. Okay. And that is what we are doing here. It will take a bit of time, let us see. Okay. So, that is what you see here. Okay. So, below you have the same information. This is the genomic location in chromosome 3R. This is 24 around 24 MB. This is where our gene is 39139. What you see interestingly is that, okay, for the control, right, the counts are much lesser compared to the histone ChIP-seq experiments. So, this is what is expected, because histone ChIP-seq experiments, they enrich for specific regions, right, because we have an immunoprecipitation step. So, only those regions that are bound by specific transcription factors or have specific histone modification, they will be represented, over represented in the sequencing library. Okay. What is also very interesting is that these peaks that you see, this is kind of like that count distribution, the distributions that you see, they look pretty similar across these 2 replicates, right, replicate 1 and replicate 2, right. So, this is actually very reassuring, right. So, both replicate experiments, they are giving us similar results. Okay. So, and this is where we also see enrichment, right, in these regions. Okay. But then now we have to call the peaks, right? This is great, right? We can see the counts, etc., across these different regions, and around the gene we can see, okay, just upstream of this gene we do not have much of this histone modification, but downstream of the gene and along the gene body we will see some of this enrichment for these reads. Okay, so once we have done this, now we want to do the peak calling, and we want to also overlay those peak calls on this plot. Okay, so this is something that's now we are going to do. Okay, so the first step now is the peak calling. Okay, so this is something we'll have to do, and we'll first do the peak calling using the Max3. Okay, so this is the tool that we are going to use. Okay, it's called Max3, right? And here we have again the page for this tool. We have all the details, okay, and how to install, etc. So you have installation guide here. How do you install this for different versions, right? What are the tools? What are the requirements that you need? How you can install them? This is a Python based tool, and there are different ways to install this. Okay, so this is something you can look at, right? So what I'm going to do is I'll simply go into the commands that we can use, and the first command that you see is the Max3 call peak. Okay, so this Max3 call peak, again it tells us, right, how we have to run this, right, and what are the essential command line options we have to mention. Okay, when you run this in Linux or Unix, we have to use these options -t, -c, etc. So all these are elaborated here very nicely, so please feel free to go through this man. Okay, so the other way is also through the command line to check, right, how this Max3 will work. Okay, so let's now move on to the Linux command line that we have already seen, and so I have already installed Max3 in here. Okay, so let's see. Okay, yeah. So Max3 call peak -h. So in this system, it's Max3 is already installed. There are a lot of steps. Please look into the installation process. So when I say Max3 call peak -h, it's kind of gives us a help, right? It tells us, right, what the options are and what these options mean. Okay, so this is something again described in their web page, but here also you can see this. Okay, so how we run this is how we run this. Okay, Max3 call peak, we can specify options -t file, right? This is the treatment file, right, or the target file. So this is the chipsec experiment that we have done. Then we have the -c, which is the control file. Okay, for that, now these are the options that you have to give. Now the format, right, this is the format of the tag file, right, auto, bed, eland, etc. So we have to so if you give the auto option, then this program will automatically decide, right, which mode it is, but we can specifically mention this this is in bed format, right? We are in bed format and not in any other format. Then we have the -g size, right, genome size. So this genome size can be anything, but we can use the shortcuts, right? Here is mentioned HS, DM, C elegans, CE, etc. For different organisms, we can use this. If we do not use anything, the default would be the HS. Okay, so this is something that you have to keep in mind. The -s we don't need to mention. This is usually not set, and -keep duplicates, right, whether you should keep duplicates, etc. Again, depending on the experimental process, we will not touch that. Okay, these are like the main options that we are going to use in our analysis. Okay, and then you also have something called let me see where we have there is a no model option, right? As I said, I'll describe what this no model is, right, whether or not to build a shifting model, right? So this Max will not build model, right? By default mean shifting size is 100, right? But if we are comparing, so as you can see here, it's highly recommended that while you have many data to process, you plan to compare different conditions, right, differential calling, we have to use this no model option, right? So we do not want to use no model option. So if you want to go into the differential analysis, right, across different conditions, then we should use this minus no model. Okay. So, that is why we are using minus no model, although we will not go into the differential analysis, but this is something that we need to keep in mind. Okay. So, now let us run the files, right. So, the file options that we have to mention are given here, right. As you can see, the maxical peak minus T, the sorted from bed, right. This is the bed file. This is where we have done the ChIP-seq experiment, and this is the minus C, which is the control file. Okay. Then followed by that, we have the minus F bed. This is the bed format. We are giving our data in bed format. Minus G, we have the Drosophila genome, right. So, the genome size should be determined for Drosophila melanogaster. We are using this minus no model, and then we have the output file name minus N H3 K36 Me3 R1. Okay. This should be the output file. Okay. And let us run this now. Okay. Okay. So, I am running this, and it will generate this peaks, right. It will generate this peak file, which we can then load in here using in the R program. Okay. So, it is running here, as you can see, and there are again it will take a bit of time. So, it is saying how many reads have been processed, and then these reads. So, after processing, it will generate this peak information, right. It will do the peak calling. Okay. So, it will do this for first file, and then we will run the second file as well, and it will generate the output in bed format. So, how they will look like is this HCK36 Me3 R1 replicate 1 dot bed. Okay. And similarly for this one, it will be replicate 2 dot bed. Okay. So, once we have those replicate bed files generated, we can load them in R here. Okay. And then we can overlay these peaks on top of these tracks that we have already generated. Okay. So, we can generate those tracks where we also have this peak information represented. So, so one thing we should realize, right, this is where we have the bin data, right. We are kind of looking at the coverage of reads around these regions of the genome, and then on top of that we will overlay the peak calls. Okay. Okay. So, let us see if the process is complete, because there are a lot of reads around here. So, you can see the number of reads that are being processed. Okay. So, this is going to take a bit of time. Okay. So, I think this is more or less done now, and well, once these bed files are generated, we will do the same thing again. We will choose only chromosome 3R. Okay. We do not need all the chromosome data, we just need the chromosome 3R data. Okay. So, this is what we are going to use. Again, this awk command, right, AWK, and then if this is chromosome 3R, then only those we can write in a separate file. Okay. Okay. So, this is where the peak calling is happening. Without going into the algorithm how this works, if you are interested, of course, please feel free to take a look. Okay. So, what you can do also in the meantime. So, this is more or less done. It give us like the final results. So, it is writing those I think results here, which you will be able to see in that folder. Okay. So, here you will see this the folders. In this folder, we will see the output file. Okay. Okay, it's taking a bit of time. Okay, so in the meantime, I think we can look into this, right? What we are going to do, the next steps. So then we can move on with that one. This is done. So once we have the peaks, will import those bed files, right? This peaks chromosome 3R dot bed, and then we'll define this peaks track, right? So as before, as we have seen here, we have defined this control track. We had defined this H3K36Me3R1 track. We then defined this H3K36Me3R2 track and all those things, right? So similarly, we'll define now the peaks track. Okay, so we have this peaks 1, right? For the replicate 1, we'll define this peaks 1 track, right? From this peak, after the peak calling is done, and then we'll define the peaks 2 track, which will load the peaks 2 information, right? And again, as you can see, right, we can we'll give this genome information, the genome version TM6. We'll have to give the name, right, that will be displayed in the plot when you draw these peaks, draw these tracks in the genome. Okay, and then we'll use the same plot tracks function, okay, where we'll have this control track H3K36Me3R1 track, peaks 1 track H3K36Me3R2 track, and then peaks 2 track. So we just simply add this along with the MET-L3 location and the genome axis track, right? The location again is around this gene, right, because this is what we are interested in. Our transcript annotation will be symbol. This is the ensemble gene ID that we have stored, and then we should see type histogram, the font size, etc. Okay. All right. So let's wait for this to finish, right? And this is where is actually calling peaks here. And then we have to also do the second one. Okay. So in the meantime, we can also run the replicate 2, right, because it will take a bit of time. The similar command, right, we need to give the actual sample name, and then we give the control name followed by this -f option, -g option, -- no model, and the output file name. This is replicate 2, so we have R2. Okay. So this first one is now done, okay, and the second one is running here. So for the first one, we can start choosing only the chromosome 3R, right. So as you can see, this is the command we'll use. So okay, before we do that, I just want to show you, right, this call will generate a lot of these files, right. So we have, for example, you'll see this here. You have this H3K36Me3R1 peaks.narrow peak. We have H3K36Me3R1 peaks.xls. We have summits.bed. So we'll use this summits.bed file to actually choose only chromosome 3R. Okay. So this bed graph file. Okay, again the awk command, awk command, and we're choosing only chromosome 3R from here. Okay, so this is actually pretty quick because these peaks are much less in number. What we can do, we can look into this H3K36Me3R1 summits file, right. This is the summit file. This is the score we have. This is the chromosome information, location, and peak 1, etc. These peak numbers are defined. But we can also look into only the 3R chromosome file, right. Okay. So only chromosome 3R information is present, right. Here the peaks are given and the peak signals are given, right. What is the score of this peak? Okay. So the location of the peaks in chromosome 3R are given. So we'll do the same thing when we get the R2 peaks, right, the second peak. So this is running. So in the meantime, what we will do is we can load the first peak, okay, and we can see how the visualization will look like. Okay. So for the first peak, this is done, right. So HTK36ME3R1 peaks, chromosome 3R, only that information is required. So we are loading here, okay, and so here we have loaded this information in the peaks file. What we have to do is we have to define this peaks1 track, right. Again we need this annotation track, and we need this peaks information, the genome version, the names that should be displayed, chromosome 3R, shape, what should be the shape box, and then you have to fill color, this what should be the color of these boxes, right, that would be inside, and the size of this box, okay. And this is the peaks1 track that we have defined, okay. Now once we define this, we can go ahead and visualize with the peaks1 track, and then once peaks2 is done, then we will do the peaks2 track, okay. So for plot 2 here, okay, okay, up to this track, whatever data we have, we do not have the peaks2 track information yet, right, because the program is still running, but you can look into the peak1 track, okay. So whatever information we have, we put this inside this C, right, we are creating this combined data, right, and then we are just visualizing this here, so with this data frame, okay. The rest of the parameters, right, they remain the same, whatever we have discussed, okay. So it's running, and we'll get this, and at the same time we can check, okay. So it's also running towards almost there, and once this is done, we can add this to this track information as well, okay. Okay, so this is done, okay. So as before, we have the count data for the control, so we can actually zoom in a bit more, as you can see clearly now. So the top one is control, right, we have the count data for these beans across control. As you can see, there is no significant enrichment, so that's good. Then we have the replicate 1 experiment for the specific histone modification, and you have see this read count enrichment for specific regions, okay. And then you see the peak calls, right, these peak calls are given in this blue color here below, right, as you can see these peak calls are here, okay. We have not done the peak call, we are not done with the peak call for this replicate 2. Once we are done, we will also add these peak calls below, right, below this data track. And then we have the annotation, right, where these genes are. As you can see, our gene of interest is here, okay. All right. So this is something that we are going to do for the other replicate as well once this is done. Followed by that, okay, so once this is done, we can also look for something called peaks overlap, right. What are the overlaps between these peaks across these two replicates, and we can also calculate this unique overlap, okay, and we can use this Venn diagram library to actually plot this pairwise overlap, okay. So this is something we will do once we have this peak information, and then finally we can also have the common peaks track, which we can also use on this visualization part, okay. So let's wait for this run to complete, right. So this is something that will take, and if once this is done, this is something, the next steps that could be is whether you have any of these peaks is overlapping with the promoter regions of specific genes. So this is something further analysis that can be done, which we will not do in this class, but this is something, this is the code that we can use if you are interested in looking into the location of these peaks in the promoter regions, right. As you probably understand, why we are interested in promoter regions is because these histone modification marks, they can actually they impact the gene expression pattern, right. So most of these histone modification marks in the promoter, they can influence transcription factor binding, so they can influence gene expression. So that's why we are especially interested in looking at the position of these peaks in the promoter region. Okay, so once this is almost done now, the second replicate peak call, and then once this is done, we'll load this here in R. Okay. After we have of course chosen only the chromosome 3R results, right, and using this AWK command. And once that is done, we will load them in R, and then we will do the similar processing where we will see the peak for the second replicate as well. Followed by that, we will simply calculate the peak overlaps, right, how many peaks are common across these two replicates, and then we will actually draw a Venn diagram. Okay. So, let us wait for these results first. Okay, so this is done. Now we will choose this only chromosome 3R. Okay, that is also done, and we can now go into the second peaks, right. For replicate 2, we have loaded this, and similarly we will define the peaks to track. And now we can put everything together, and we can put in the visualization part. Okay. And then we can find these overlaps between these peaks, and we can use a Venn diagram, right. Okay. So, as you can see, now we have these peaks for the second replicate as well, and as you can see they are kind of very at very similar locations, right, in both replicates. So, that is good, but maybe they are not exactly same location. So, this is something you will see. Peaks overlap, this is the function find overlaps, and it is about 68 of them are common up out of a large number, right. So, this is something that is there, because their exact location may not be same, but they are actually pretty close to each other. So, that is actually good, right. The other thing we will do is use this Venn diagram library, and we will use this draw pairwise Venn function, where the area 1 will be the number of peaks that we have for the first replicate, and the area 2 will be the number of peaks that we have for the second replicate, and then cross area will be this unique overlap, right. So, this unique overlap will calculate, okay, using this peaks overlap function, right. So, peaks overlap variable that we have now just calculated. Okay. So, what we are doing is looking at length unique query hits peaks overlap, right, how many unique are there, right, and then out of this length unique subject hits peaks overlap, right. So, subject hits is here. So, how many unique are there, and out of those how many are common, right. So, so that will give us the unique overlap. Okay, unique overlap 68, as I said, right, this is kind of given here, and now we can plot this Venn diagram. Okay, so Venn diagram is actually generated on top of this. So, what we will do, we will first close this, and okay, this is the Venn diagram where you have replicate 1, replicate 2. We can have this number of peaks that have been identified in replicate 1 total, this, and then we have the 68 that are common across these two replicates. Okay, and this is something again we can do here, common peaks, right, and this is the reduced subset by overlaps list peaks of the replicate 1 and the peaks of the replicate 2. Okay. So, we are getting the common peaks here, okay, and you can see the sequence names, chromosome 3R, the ranges, etc., and the score. Okay. So, what we can do again is we define this common peaks track, and we can add to that plot, okay, and we can do the final plotting here. Okay, so, this is something we have used. We just kind of added another track here. You can see this. Okay, this annotation track here, this green color, this is the common peak. But as you can probably realize that there are many of these peaks are actually very close to each other. So, maybe we have to tweak this analysis and maybe look into like how close these peaks are to each other from their location in here. Okay, so, I think with that we are done with the hands-on part for the first data set, and this is what we have achieved in the last few classes, including this one. We have actually run the full pipeline for the chipsec data analysis, starting with BAM file, and then we have processed through binning, then peak calling, and the final visualization, where we could see this enrichment of reads as well as the peaks. Okay. And we have used several R bioconductor packages in this process. So, again, I have introduced at least some of these packages and what their functions are. In the next class, we will be taking the other data set and will visualize the results that are there. Thank you.